We learned a multitude of topics in genetics lab this semester, and today we're going to talk about recombination and genetic mapping. So, what is recombination? Recombination is an event that occurs exclusively during meiosis, specifically during prophase 1. It is when homologous chromosomes pair up and exchange genetic material through a process known as crossing over. Once this process of crossing over is complete, the chromosomes of the organism undergoing meiosis now possess a unique combination of genes that differs from the parental and maternal DNA. This process of recombination also eventually results in something called recombinant genotypes and phenotypes. These are phenotypes and genotypes that differ from the parental genotypes and phenotypes due to this new unique combination of genetic material within the chromosomes of the offspring as a result of the crossing over process. We saw an example of this during our Drosophila module, when we crossed a heterozygous wild-type fly with the homozygous recessive onyx shorty fly. There were certain instances in which offspring just possessed the onyx mutation or just the shorty mutation, thus making them both examples of recombinant phenotypes and genotypes, because they differ from the parental ones. As the field of genetics has continued to evolve over the decades, we are now able to quantify the distance between genes on a chromosome through a process known as genetic mapping. Genetic mapping is essentially exactly what it sounds like. We are mapping the different genes on a chromosome based off their inheritance patterns in relation to each other. How do we do that? Well, first we calculate something called the recombination frequency. The recombination frequency is essentially quantifying the proportion of the progeny that are recombinants. To calculate this, all one has to do is divide the number of recombinant offspring by the total number of offspring. Then, to calculate how far away the two genes in question are from each other, one multiplies the recombination frequency by 100. Doing this gives the distance between the two genes in the genetic mapping units, centimorgans. 1 centimorgan is equal to a 1% chance that two markers on a chromosome will become separated from one another due to a recombination event during meiosis. So with this final distance, we are able to quantify how far apart the two genes in question are from each other. In other words, the smaller the distance between two genes on a chromosome, the less likely that they will be separated due to a recombination event during meiosis. Alright folks, that's all we have for today for recombination and genetic mapping. It's really cool to see how far science has come and how we're able to do such advanced work now when it comes to learning about different genomes. Okay, DNA rock stars, keep rocking on.